is Mr. Ahmad Tihani. He will talk about the synthesis and biology for evaluation of novel rhodamine-based structures with antiviral activities. Please go ahead.
Thank you. Good afternoon, everyone. Hajan, my colleagues, ladies and gentlemen. Today, I will share my ideas.
Hold on, Ahmad. You have not shared the screen on here. Okay.
How do we make this one? Yes. The figure here.
Yep. Move it up.
Okay. Good afternoon, everyone. Ajan, my colleagues, ladies and gentlemen. Today, I will be sharing my understanding on the paper Synthesis and Biological Evaluation of Novel Rhodamine-Based Structures with Antiviral Activity towards HHV6 virus. In this presentation, I will start by introducing us to HHV6 virus and the rhodamine scaffold. Then we will move on to the methods, where we will talk about the synthesis of the rhodamine scaffolds, the invention and control of the virus. Then we will move on to discuss the results, and then we will draw conclusion, then acknowledgement. HHV6 stands for human herpes virus. This virus is an enveloped virus, and it has human being as its primary host. HHV6 virus are basically two. We have HHV6A and HHV6B. The major differences between these two types, is that they have different engagements of cell receptors. HHV6A basically affects all nucleated cells, while HHV6B affects basically T cells. HHV6B has been attached to be the primary agent for exanthema subitum, which is also known as Rosella infantum. This is a disease that affects infants, and it has symptoms ranging from fever, cough, appetite, and so on. HHV6A basically does not have a primary cause, but it has been associated with multiple sclerosis and Alzheimer's disease. This could be an effect of HHV6B, because this is a neurodegenerative disease of a normal brain. Then the Rosella infantum could have effect on an infant like this. Unfortunately, at the moment, there is no approved compound yet for the treatment of HHV6 virus. But basically, because HHV6 virus is an enveloped virus, for all enveloped virus, before they can have their virulence, they will have to attach tightly to the cell receptors. Now, which will lead to the injection of the cells, and the cells can also infect another cells to form cell-cell fusion. So basically, this cell and virus fusion can be stopped by the rhodamine-based structure, which is the main focus of this paper. The rhodamine-based structure has a rare part, that is, it has the hydrophobic part and the lipophilic part. The hydrophilic part can be modified, in which we will have different derivatives, and the lipophilic part also can be modified. This is done in order to have the best and the safe compound for the treatment of HHV6 virus. This rhodamine-based structure scaffold works basically by one, disorganizing the viral envelope. The envelope of the virus will be disorganized, and thereafter it will lead to the inhibition of the virus cell fusion. That is, if the viral envelope has been disturbed, has been already disorganized, there will not be attachment of the virus to the cell. And by so doing, we will avoid the release of nucleocapsid from the virus to the cell. So there won't be infection. Now to the synthesis and the structural activity relationship of the compound, we need to synthesize the rhodamine core. The synthesis occurs by using acetylbromide together with a primary amine in carbon disulfide. This reaction takes place in one-pot reaction to get the rhodamine core. So different derivatives has been synthesized here from 4A to 4F. The primary amine is used basically in order to have different derivatives of the rhodamine core, and we have moderate to good yield of the rhodamine core here, which forms the lipophilic part. Now we have to synthesize the hydrophilic part too. The iodo derivatives react with the 5-membered heterocycle compound here through palladium catalyzed Suzuki reaction to form the products here, 7A to 7E. Thereafter, they are being hydrolyzed using lithium hydroxide to form the hydrophilic part with different derivatives. Now we can now ask that how then can we combine the rhodamine core with the hydrophilic part in order to have the rhodamine scaffold? The hydrophilic part that has just been synthesized now can be reacted with the rhodamine core through Novanen-Gell reaction. Now to form the final product here, we have 9A to 9F. They are different in the value of the R here. So we do all this to have the best and the safe and the most active derivatives. Also, we are able to synthesize more 4 compounds, making 10 in total, by modifying the hydrophilic edge to get 10A to 10D. So 10 compounds in general has been synthesized. The compound synthesized works on the virus because this is the phospholipid bilayer of the virus. The compound will be able to work on the lipid layer of the viral envelope, and by so doing, we'll be able to achieve virolysis at the end. The damage that will be done to the cell depends on the derivatives of the compound that is being used. Basically, HHV6A and 6B are being injected. It's used to inject cell, and the treatment is through rhodamine-based compounds. The injection was done at different periods, like 24 hours, 2 days, 3 days, and 7 days after injection. The cytotoxicity evaluation was carried out, and the antiviral effect analysis was also done. The cytotoxicity of the compound is being assessed through MTT assay to check the cell viability at different concentrations. The 6 hydrolipophilic tail that has been synthesized are being tested against the control. At 100 micrometer, we can see that there is a sharp decrease in the cell viability, while at 10 micrometer and 1 micrometer, the cell viability remain intact, which means that at 100 micrometer, we will not be able to use we will not use 100 micrometer. The antiviral activity is being tested at 1 micrometer and 10 micrometer. Of course, we have nullified the 100 micrometer since there is cytotoxicity. Now for HHV6A, you can see that it's only 9E and 9F that show antiviral activity because there is reduction of the viral load. And the same thing for HHV6B. At 10 micrometer, 9A also join 9E and 9F. That is a reduction of the viral load, but there is a reinvention at 7 days post. There is a reinvention at 7 days after. So 9F does not really show, it's like a suppression. So we can see that 9E has total suppression of the viral load against the HHV6A and HHV6B virus. The modification of the hydrophilic head at 10 micrometer, when we check for the 4 compounds, you can see that 10C has there is sharp reduction of the cell viability, which means there is at 10 micrometer and 1 micrometer, the cytotoxicity of 10C is too high. So it will not be considered for that. Now for 10A, 10B, and 10D, we check the antiviral activity, and we can see that for HHV6A, only 10D showed reduction of the viral load at 24 hours post-injection and 48 hours post-injection, and there is a reinjection at the end. So basically, the hydrophilic head for all the compounds show reinjection. Now we can conclude that there is an increase in the amphipacity of 9E derivatives because it has less toxicity and it is the most active. The rhodamine scaffold can be regarded to as fusion intermediate inhibitors because they promote they stop the release of the nucleocapsid of the virus into the cell. The rhodamine scaffold help in the blocking of the virus cell fusion. And also, the different derivatives were able to be synthesized through substitution and bioisosteric replacement of the hydrophilic head and the lipophilic tail. Again, the 9E derivatives has the best activity with less toxicity. My special thanks to my advisor and the seminar committee. Thank you all for listening.
All right, thank you, Ahmad, for a nice talk. Now it's time for Q&A. Yeah, 15 minutes. Students in the audience, please type your code and your name in the chat box. And you can turn on your microphone to ask a question. Right, we have the first person to ask, A107, Nafisa. A107, please turn on your microphone.
Hello, good afternoon, everyone. I want to ask that what is the aim of what is the importance of the research? What is the importance of the research?
Okay,
Hello,
thank
can
you.
you hear me?
Yes, I can hear you.
Okay, that's it. What is the importance?
The objective of the research is to synthesize rhodamine-based structure that will have antiviral activity towards HHV6 virus.
And what is that HHV6 virus? What is the effect on human being?
I mentioned that we have two basic types, the HHV6A and HHV6B. The HHV6A has been associated with Alzheimer's disease, and it can also cause infertility and also multiple sclerosis. While HHV6B is attached to Ricella infantum.
Okay, thank you.
You're welcome.
Okay, next is 0118, Ahmad.
Hello, Ahmed. Can you hear me?
Yes, I can hear you.
Nice for your good presentation. And can you go back to your slide number 11?
Yeah.
Yes, this one. You mentioned that in this paper they made 9 derivatives of your compound rhodamine. So I have a question in biological activity. Could you please give us information which compound will give the a good activity?
Yeah. From this slide, we use the derivative that has been synthesized here at different concentrations. We use it at 100 micrometer, 10 micrometer, and 1 micrometer. And we discovered that at 100 micrometer, there is too much of a cytotoxicity here when subjected to MTT assay. Then it means we will not be able to use it at 100 micrometer because it is not safe. So we only consider at 10 micrometer and 1 micrometer for the antiviral activity.
Okay. Could you please mention which compound will give the good activity
Yeah.
in these nines?
From here now, at 1 micrometer, there is reduction of the viral load against the HHV6A and HHV6.
Sorry, Ahmad, can you switch the screen? Because now it's a different page. And stop share and then share again another screen.
This
The left one. Okay, it's good.
Okay, I was explaining that at 1 micrometer for HHV6A and 6B, the 9E and 9F showed reduction of the viral load, which means they have antiviral activity at that concentration. But at 10 micrometer, which is the almost the best here out of the three, there is complete reduction of the viral load for 9E. So 9E is the best and the most safe.
Yeah, I understood from this slide. Thank you. I have one more question. In slide number 10, you mentioned, can you go back to slide number 10? Yes, in this slide, you mentioned the process of the compound which injure to the virus. I wonder about this. Can you briefly give information about this process? How is it worked?
Please, can you come again? I cannot understand the question.
In this slide, you mentioned the compound, the compound insert to the virus, and they will do the process. So, can you briefly give us information about this process? I didn't understand.
Okay. The HHV6 virus is an envelope virus. And what that means is that for it to have virulence, to infect any cell at all, it has to attach tightly to the cell. Now, the compound that has been synthesized based on rhodamine structure has the ability, because it has lipophilic tail, and majorly the viral envelope is made up of phospholipid bilayer. So it has the ability to oxidize the lipid. So by oxidizing it, it can disorganize it. So you can imagine this virus without, like, it's like you have broken its shell, so it does not have any ability to infect any cell again, as it is here now.
Yes,
So,
thank you.
So
I have no more question. Thank you. Thank you for your nice presentation.
You're welcome.
So what do you mean oxidizing the phospholipid bilayer?
Yeah, it's more of dissolving the phospholipid bilayer.
I think it's called destabilizing. You see the middle one, the middle picture is the destabilization of the envelope structure of the virus.
Yeah, the destabilization was being able to achieve through the lipid oxidation.
Yeah, I'm not sure about the oxidation I've never heard of this thing in the virus.
So do I. I am not sure about the oxidizing destabilization. I think it's the word.
Yes, I agree, ครับอาจารย์สนอง So do you know what is the oxidation reaction or the oxidation process? So it involves like some electrons, donation and electron like charge changes. But for your compound, if you have no evidence that it can undergo like oxidation or reduction with the viral membrane or the viral envelope structure, you shouldn't claim that. So just the destabilization, I think that's the appropriate word. Okay, let's go to the next student first. Or อาจารย์พุทธรักษา has some more question about this.
Okay. I still understand about the polar and the non-polar, the lipophilic head that you would like to modify. Can you go back to slide 6 or 7, something like that, and can you explain to me again that, yeah, what you try to do in the polar head and the lipophilic tail on your compounds and to test for the bioactivities?
Thank you. The modification of the hydrophilic head here, we can only modify the carboxylic acid here and the hydroxide here. So we can exchange the position of these two. That is the modification of the hydrophilic head. The modification of the lipophilic tail is by using different derivatives.
And then what you're trying to do, like what objective to you want the long change, or what happened if you modify that polar head and lipophilic tail? What you expect? I mean, when you modify into several compounds of your modification, I couldn't match the slides, previous slide and the slide that you have many derivative one.
Yes, the derivatives are being synthesized to be able to have different compounds to choose from.
Yeah.
So we did the modification of the lipophilic tail to get the 6 compound here.
Yeah, and I mean, what did you modify into the longer chain? Like increase the chain, or you add some group into? I'm not sure. I'm not the synthesis person.
The chain is increased too. Then we also change.
So the in series 9A to 9F.
Yeah,
you change
9A to 9D. That is extension of the chain, and 9E to 9F. That is a change in the configuration. We have R and S configuration for 9E and 9F.
Okay, and what property that you think you expect from them? That when you have the different side chain,
basically,
something like that. I still don't get the idea when you do the modification for each tail and head.
Yes.
Can you summarize into like a short sentence that I can understand what the purpose of your work?
The idea is basically to synthesize a derivative that will have better interaction with the viral envelope. So we can see that majorly the lipophilic tail is the one that shows the highest antiviral activity, possibly because the phospholipid bilayer of the virus is have better interaction with it.
Because you already come into the structure, so I have the questions. Why it's supposed to be rhodamine structure? Can you do something else?
Why
Can
is
you-
it supposed to be?
Why is this, why is
Use rhodamine structure.
yeah, why you use the rhodamine as the base structure?
The reason basically is because the rhodamine scaffold have both the hydrophilic and the lipophilic part.
So I think the question that Professor Somodi is asking is that like how do you come up with this structure, this core structure of the rhodamine, right?
Okay. The rhodamine scaffold has been used in the past for antiviral treatments.
Okay, it has been used before.
Been used
I
in the
see.
paper. They mentioned that it has
Okay.
been used against influenza A virus
Oh,
and also HSV virus too. Yeah.
do we have rhodamine in nature?
In nation, no. I don't think we have rhodamine in my opinion. But it's always been synthesized.
Usually it's synthesized.
Okay, let's go to the next student to ask 0114, Mutita.
So my question is very similar to what Ajahn just asked you, because from the, can you go to slide 11